back to this biology class 10. Myself, Ajay Kumar Mishra. Dear student, in my last class, I have taught you various types of nutrition, which is one of the life process in the living organism. Today, I will teach you one of the type of autotrophic nutrition, that is photosynthesis. Photosynthesis is made up of two words. Photo means light, synthesis means production, that is production of food in the presence of light. So photosynthesis is a biochemical process during which green plants manufacture organic foods from carbon dioxide and water in the presence of sunlight inside chlorophyll containing cells. The full photosynthesis process can be represented by an equation which is as follows. 6CO2 plus 12H2O in the presence of chlorophyll and sunlight gives C6H12O6, that is glucose, 6O2 plus 6H2. Dear students, if you see this equation, you will find that water molecule is used as raw material, but water molecule is also produced during this process. So some of the people write this equation in a short form like 6CO2 plus 6H2O in the presence of chlorophyll and sunlight gives one molecule of glucose, that is C6H12O6 plus 6 molecule of oxygen. But this equation is technically not correct, because this water molecule which is produced is entirely different from the water molecule which are used as raw material. Actually, this water molecule is produced by breaking down of carbon dioxide molecule. So this equation is correct. During the photosynthesis, glucose is produced as main product, while oxygen and water vapors are released as byproduct. During photosynthesis, four major components are required, that is carbon dioxide, water molecules, chlorophyll and sunlight. Carbon dioxide is absorbed by the terrestrial plants from the atmosphere, mainly through stomata of leaves. Water molecule is absorbed by the roots of the plant and transported it to the leaves through stems. Chlorophyll is mainly present in the leaves and sunlight is available during daytime. Apart from that, plants also obtain nitrogen, phosphorus, sulfur, iron, magnesium, etc. from the soil. Actually, nitrogen element is used in the synthesis of protein inside the plants. These all are together called raw material of photosynthesis, that is carbon dioxide, water, chlorophyll, sunlight, nitrogen, phosphorus, iron and magnesium. These are raw materials of photosynthesis. Photosynthesis actually mainly occur in the green leaves of the plant, but it also takes place in the green stems and the floral parts of the plants. So let us study the structure of leaf. If you see the cross section of leaf under the compound microscope, you will find three main regions. Upper epidermis, which is covered by waxy layer called cuticle. Below it, there is a mesophyll region. Actually, this is the main region of photosynthesis. This mesophyll cell is of two types. One is palisade cell and other is a spongy cell. Palisade cells are cylindrical in shape and compactly packed, while mesophyll cells are spherical in shape and loosely packed. Chloroplasts are mainly present in this palisade cells. You can see the chloroplast as dot-like structure. But spongy cells also contain chloroplast. Chloroplast is the cell organelle which contains a green pigment called chlorophyll. During photosynthesis, three major events take place. First of all, light energy is absorbed by chlorophyll. Second, light energy is converted into chemical energy, and water molecule is split into hydrogen and oxygen. And third event is the reduction of carbon dioxide into carbohydrate. The glucose molecule is this glucose molecule is the simplest form of carbohydrate, which turns into complex carbohydrate, that is starch, and starch is stored in the form of reserved food in the different parts of the plants. During the photosynthesis, we have seen that the major components are required, that is chlorophyll, carbon dioxide, sunlight, etc. So let us do an activity which shows that chlorophyll is essential for photosynthesis. For that, we will take a potted plant. We will take a potted plant having variegated leaves. Actually, the leaves are generally green in color, but some of the leaves have many colors. Like you can see this leaves, which is a money plant leaves, having green colors and yellow colors too. In the croton leaves, you will see different colors. Such types of leaves which have different colors are called variegated leaves. So we will take a potted plant having variegated leaves. We put this potted plant in a dark room for 3 days so that starch present in the leaves get used up completely. After that, we put this potted plant under sunlight for 6 to 7 hours. After that, we pluck one of the leaves from that potted plant and we mark the green parts of the leaves and sketch it under white sheet of paper. Then we wash this leaf with warm water in order to remove dust, dirt, etc. from the surface of the leaf. After that, we insert this leaf in a test tube containing alcohol and put the test tube in a water bath for heating. Here we heat this apparatus gently, not strongly, because alcohol present in the test tube may catch fire. Why? Because alcohol is highly inflammable substance. When the water in the water bath becomes warm, the alcohol present in the test tube starts boiling. After few minutes of boiling, you will observe that the leaf become discolored. This is called bleaching of leaves. After that, you take out the leaf gently outside the test tube and again wash it with warm water. Then you put it in a petri dish containing iodine solution. After a few minutes, you will find that the green parts of the leaves turns blue-black, while the non-green parts remains colorless. Because in the green parts, chlorophyll was present, so in this part only starch formation takes place and this portion becomes blue-black in the presence of iodine. Since no photosynthesis occurred in the another part, so no starch formation occurs and this will remain colorless. This shows that chlorophyll is essential for photosynthesis. Let us talk about another essential components which is required for photosynthesis, that is carbon dioxide. For that, we will again study the internal structure of leaves. At the lower epidermis, you will find minute pores, which is called stomata. These pores are microscopic in nature. It means that you cannot see these pores on the surface of the leaf with our naked eye. You can see it under the microscope only. This stomata is generally present on the lower surface of the leaf, but it also present on the upper surface of the leaf. Through this stomata, exchange of gases takes place during photosynthesis. It means carbon dioxide is taken inside and oxygen produced during photosynthesis is released out through this stomata. But through this stomata, another important process also occurs, that is called transpiration. Transpiration is the process of loss of water in the form of water vapors. So, for the photosynthesis process, plants have to lose water. In order to conserve water, many plants close their stomata during night times and stomata becomes open during daytime. This opening and closing of stomata is a function of guard cell. Guard cells are kidney-shaped cells which surround the stomatal pore. You can see the guard cell, this kidney-shaped cell which surround the stomatal pore. These guard cells absorb the water from the neighboring cell during daytime. Actually, the outer lining of guard cell is very thin and inner lining of guard cell is very thick. So, water put more pressure on the outer lining of the guard cell. As a result, guard cell bulges out and stomatal pore becomes open during daytime. During night time, water molecule is diffused out from the guard cell to the neighboring cell, due to which guard cell shrinks out and becomes flaccid. As a result, stomatal pore closed during daytime. In this way, opening and closing of stomata takes place. Let us now do an activity which shows that carbon dioxide is essential for photosynthesis. For that, we will take two potted plants of the same height and put it under dark room for 3 days. Put it under dark room for 3 days so that starch present in the leaves gets used up. After that, we put this potted plant in a glass plate separately and we take potassium hydroxide in watch glass and put beside one of the potted plants, and then we cover both the plants with bell jar and apply Vaseline at the lower portion of the bell jar and the glass plate so that both the apparatus becomes airtight. Potassium hydroxide present in one of the bell jar absorb all the carbon dioxide present inside it. We put both the apparatus under sunlight for 6 hours. After that, we pluck one leaf each from the plants and go through the starch test as we have done in the previous activities. That is, putting the leaves in a warm water, then we put the leaves in a test tube containing alcohol for boiling, and after boiling the leaves become this color. Then we put the leaves in iodine solution. At the end, we will observe that the leaves which was plucked from the potted plants having potassium hydroxide will not turn blue-black. It remains colorless, while the other leaves turns to blue-black. Why? It is because these plants will not get carbon dioxide and these plants get the carbon dioxide. These activities prove that photosynthesis, for the process of photosynthesis, carbon dioxide is essential. After that, we will discuss another activity which shows that sunlight is essential for photosynthesis. For that, we will take potted plants having broader leaves and we put it in a dark room for de-starchation. It means that during this process, the starch present in the leaves gets used up. After that, we put this potted plant under the sunlight and cover one of the leaves with black paper with the help of clips from both the sides. After 6 to 7 hours, we pluck the leaves, that covered leaves from the plants, and remove the black paper. And then we go through with that leaves for starch test. At the end, what we see? We see that the uncovered portion of the leaf will turn blue-black, while covered portion will not turn blue-black. You can see that this is the leaf, this portion was covered, and this portion was not covered, so this portion turns to blue-black, while the covered portion remains colorless. This shows that light is essential for photosynthesis. At the end, I will give you an assignment having 10 questions. First question is, where do plants get each of the raw materials required for photosynthesis? Which cell organelle is the site of photosynthesis? What is variegated leaf? Prove experimentally that carbon dioxide is necessary for photosynthesis. Next question, proved experimentally that chlorophyll is necessary for photosynthesis. Next question, proved experimentally that light is necessary for photosynthesis. Next, explain the process of opening and closing of stomata. Next, draw a well-leveled diagram of the cross section of a leaf. Next, what is photosynthesis? Write a balanced equation of photosynthesis. And the last one is, write the major events occur in photosynthesis. So, with that, this is all about of this class. Goodbye. Please take care of yourself. Thank you.